My name is Yuning, and I'm from International Peace Trinity Child Health Hospital, School of Medicine, Shanghai Jiaotong University. And this picture is my hospital, located at Shanghai, China. I have studied the role of extracellular vesicles on male fertility for 5 years, and discovered several novel factors of the extracellular vesicles from spermatogonia. And today I'm going to talk about THY1 positive spermatogonia suppresses the proliferation of spermatogonial stem cells by extracellular vesicles. SSC is short for spermatogonial stem cells, and EVs is short for extracellular vesicles. The presentation will be in 4 parts. First, I'm going to talk about the introduction. As we all know that the SSCs are undifferentiated spermatogonia that are essential for the maintenance of spermatogenesis. They balance the self-renewal and differentiation to maintain their numbers. And this is the schematic of spermatogonia development and the molecule markers. The SSC is in the basal compartment of the seminiferous tube, which continually proliferate and produce the progenitor spermatogonia, such as A line. Subsequently become the differentiated spermatogonia, then enters the true meiosis. And it is reported that the SSC occurred cyclically in the seminiferous epithelium, which proliferate primarily in stage anus to 6 and remain relatively quiescent at stage 7 to 10, which means that SSC division pattern is cyclically regulated and involves positive and negative regulation. And current understanding the signal pathway regulated SSC self-renewal in mouse testes are influenced by extrinsic factors from Sertoli cells, which are the early somatic cells physically interact with SSC with the seminiferous tube. Sertoli cells provide growth factor GDF, FGF2, and chemical CXCL12, CCL9, bind the receptor GFIA1, FGFR, CXCL4, and CCR1 present in SSC to regulate the self-renewal through activate the PI3K, SFK, or MAP2K signal pathway. On the other hand, the differentiation of SSC were regulated by stem cell factor and reticulic acid, but it remained unknown about how microenvironment precisely controlled their self-renewal division of SSC, especially the negative regulators. Next, as we have found that the extracellular vesicle regulates the proliferation of SSC, let me first introduce the background of EVs. According to mineral information for studies of extracellular vesicle 2018. EVs as the generic term for particles naturally released from the cell that are delimited by a lipid bilayer and to large contained affection nucleus. Extracellular vesicles include exon, apoptotic body, and microvesicles. And the EVs could carry variety cargoes such as protein, enzyme, mRNA, lipid, DNA. They can be taken up by other cells depending on their origin. They have different physiological functions. Current research on vesicles in the male reproductive system mainly focus on epididymis, prostate, and seminal vesicle. The epithelial cells of the epididymis produce a population of EVs called epididymal zone. Epididymal zone fused with the sperm and transfers the protein to sperm to regulate the sperm's maturation. And the prostate also secretes the EVs, sometimes in terms of prostations. It suggests that the prostations were largely originated from the multivessel body, and suggests that the prostations might interact with the sperm in the female reproductive tract to facilitate the sperm reach the oocyte. Even though the composition and the properties of EVs have been intensive investigation from on, but the role of the EVs in testes is still unknown. Let's move on to second part, the origin of testicular EVs. When we were observing the testicular electric microsection, unexpectedly we found a large number of EVs near the basement of a seminiferous tuber. We then tracked them in the testes from different age months, 8 days, 40 days, 21 days, and 8 weeks post part 2. Use the transmission electric microscope. You also observed each in the basal compartment of the seminiferous tuber. In addition, we also found a specific structure in the cytoplasm of type A spermatogonial. Is called the multivesicle body, which is the precursor before exon release. So, we suppose that the testicular EVs might secreted by spermatogonial. Then, the immunoelectric microscope further showed that testicular EVs were detected with spermatogonial marker JFI1. They isolated the testicular EVs with a plot showed that they were detected together with spermatogonial member proteins JFRA1, THY1, CD9, and the external protein AT1 collectively. This result indicated that testicular EVs were secreted by spermatogonial. We then investigated it in different species, and the figure showed that the testicular EVs in testes were conserved among the rat, mouse, rabbit, and human. And we can see there are a large number of the EVs in the basal compartment of the seminiferous tuber, and the picture is clearly than the previous finger, which suggests that the testicle is conserved and may have an important role in development of testes. Let's move on to the third part. To explore the function of testicular EVs in spermatogenesis, we'll first separate the testes EVs by two steps, either digestion and differential ultracentrifugation. To confirm the EVs purification, samples were examined using the particle tracking analysis, transmission electron microscope, and vesicles approach. Both particle tracking system and transmission electron microscope revealed that diameter of EVs ranged from 15 to 120 nanometers, and West approach confirmed the presence of extracellular protein CD81 and CD9 in EVs, confirmed that the EVs is purified. As the testicular EVs were secreted by spermatogonial that might interact physically with SSCs to determine when the testicular EVs could be taken up by SSCs. We label the EVs with the excrement. 1 hour after exposure to testicular EVs, the EVs were soon uptaken by green SSCs, while the male feed cells did large exhibit red fluorescence. To further explore the effect of testicular EVs on SSCs, we exposed the SSCs to various concentrations of testicular EVs, as shown in figure. Testicular EVs suppressed SSC proliferation on concentration-dependent manner and reduced carbon size. These results suggest that testicular EVs suppress the proliferation of SSC in vitro. Next, I'm going to talk about the last part. THY1 positive EVs suppress the proliferation of SSC. I have tried several spermatogonial marker protein antibody magnetic beads, and only the THY1 antibody component magnetic beads worked well. So specifically separate the EVs from spermatogonial, we use the THY1 component magnetic beads to capture the THY1 positive EVs, and the West approach and the immunoelectron microscope confirmed that this approach is effective. Then, to study the effect of THY1 positive and negative EVs on proliferation SSC, we use the EDU assay, and the results showed that the THY1 positive EVs significantly replace the proliferation of SSC, and the THY1 negative EVs have no effect on the proliferation of the SSC. We next explore the THY1 positive and negative EVs on apoptosis of SSC, and the figure shows that both THY1 and THY1 positive and negative EVs have no effect on the apoptosis in SSC. In summary, THY1 positive EVs suppress the proliferation of SSC but not lead to apoptosis. Finally, I'd like to conclude today's presentation by summarizing the information that we first found the EVs in testes, and that is conserved among rat, rabbit, mouse, and human testes, and proved it secreted by spermatogonia in mouse. Further confirmed that THY1 positive spermatogonia suppress the proliferation of SSC but not lead to apoptosis in vitro, which provide insight into a novel mechanism of regulating the SSC self-renewal, that spermatogonial suppress the proliferation of SSC, used a negative feedback to regulate the SSC proliferation by secret EVs. All these data presented today have just published in Endocrinology. In here, I have to thank my mentor, Dr. Song Fei, and colleague Mr. He Wen, Mr. Gong Xiaowen, and so on for their help. And thank you for your listening. If there are any questions, I'd be happy to address them.